Welcome to the Garden Talk podcast, where we interview growers from all over the world, both beginners and experts, seeking to learn more about what they know about gardening and how they do things in their garden. Hey, what's up everybody? For those of you that don't know me, my name is Chris, aka Mr. Grow It, and you're tuned into the Garden Talk podcast. This is episode number 148. In this episode, I interview Luna Whitcomb. She has been on the podcast once before, episode 111, where she talked all about things that you can do in order to increase plant taste and smell. One of the things that she mentioned in that episode is that she uses ferments, which prompted some of you to comment on that video requesting more details. She has a ton of knowledge on this, so in this episode, we get into it. She talks about the different types of fermentation and how you can implement in your garden. If you want to see short clips of these podcast episodes, search Garden Talk Clips on YouTube. I also have a gardening channel where I show the plants that I grow. Both of those channels will be linked down in the YouTube description section below. One of my goals for this podcast is to bring free information about gardening to the general public. That being said, I'd like to thank the sponsors of today's episode who helped make that goal possible. Thanks to AC Infinity for sponsoring this episode. Check out their advanced grow kits, which include nearly everything you need to start growing indoors. They have several different size kits, and they all come with a grow tent, grow light, ventilation system, oscillating clip fan, a controller 69 Pro, and so much more. Click the link in the description section below so you can learn more about their grow kits, and the discount code MrGrowIt15 works on both Amazon and their website, acinfinity.com. Stash Blend. Stash Blend is a 2-1-5 plant additive that can be used with synthetic bottled nutrients or in living soil systems. Simply mix half teaspoon of Stash Blend into a gallon of water, then water your plants with it. Ingredients include corn steep liquor, seaweed extract, humic acid, beneficial bacteria, silica, and mycorrhizal fungi. Check it out at stashblend.com, link in the description section below, and use the discount code THESTASH. And we're back, and I'm joined with Luna Whitcomb. How you doing today?
Good, how are you? Thank you for having me back.
Yeah, second time around. First time was awesome. We got real deep into things. You're one of the more advanced growers, I'd say, of just advanced knowledge, and we had a lot of folks here that tuned in and they were just blown away by all the knowledge that you dropped that time around, and they wanted you back for a second time. So here we are for part 2.
I'm happy to be here. Let's go a little bit deeper. Let's give the people what they want.
Absolutely. So today we're going to talk all about fermentation for plants. And when I think of fermentation, I think a lot of people think of like KNF. They think of the fermented plant juice, fermented fruit juice, but there's a lot more to it than just those things. And that's what we're going to get deep into today. There's so many different ways to go about it. I know you have certain ferments that you do. So finding out what you do, when you apply them, how you make them, all that stuff. We'll cram that into this episode. We'll see how far we can get. And then should be a very knowledgeable, packed episode. Could you just introduce yourself for those that didn't catch the first episode we did together?
Yeah, sure. My name's Luna Whitcomb. I am a educator in the kind of living soil field. I do soil mineral balancing consulting. On my Instagram, I share a lot of different recipes, like the ones we're going to be talking about today, and like different approaches to things and how you can like up your game and up your soil health, your plants' a volatile organic compound profile, which is like just the smell, make things smell better, get your yields up, stuff like that. You know, I have like a Patreon. I'm a breeder. I breed, different types of seeds and stuff, and genetics that I like to work on. I've been doing that for about 10 years. I've been cultivating for about 16 years, and this is my passion. Soil and plants are my passion.
Awesome. And like I mentioned, we did do one episode together in the past. I'll definitely have that linked in the description section below. So you can, after this episode, you can click on that one and go tune into that one. Okay. Fermentation. Let's get into it.
There's so much to know about fermentation. And so you mentioned a couple like KNF inputs, FPJ, fermented plant juice, fermented fruit juice and stuff like that, which is like a mixture of a sugar, but it's just any kind of high osmotic pressure input that will pull the juices out of plant matter. And then they leave it open. You leave it open with like a coffee filter, and it'll collect, microbes specific to that profile of compounds inside of the organic matter in that juice. So like different enzymes and amino acids and carbohydrates will attract specific microbes that, want to live in that environment. So you'll collect, microbes specific to that plant matter, and will help break it down, make it available. And that's what makes it a fermentation, because a fermentation is just a biological breakdown or changing from one compound to another through biology in plant matter. So there's a whole bunch of different types of fermentations out there. Like you mentioned, the FPJ, the FFJ, and stuff like that. That's an osmotic fermentation. Okay. But we also have solid state fermentation, which is like Bokashi, where you take a dry bran and you add lactobacillus to it. We can get into the details of how to make that stuff. There's also anaerobic fermentation, which is usually used with lactobacillus, lactic acid bacteria serum. And then that's also added to plant matter, and you can add different inputs like calcium and amino acids. And then there's, an aerobic fermentation, which people often call compost teas. And I think compost tea is kind of it's the very, very widespread term used for making them, but they're just like aerobic fermentations. And I think that that's a more accurate way of describing it. So yeah, I'd love to break down each different category and like how to make them and how I like to approach it.
Where would you like to start? Which one should we kind of begin with?
So the foundation for a lot of them is going to be making lactic acid bacteria serum. And this is a practice taught in the Korean natural farming world, but it's actually goes back way before that, way earlier than Korea. It goes back to Japan and even, and to Greece. And this is a practice that was used all over the world almost simultaneously. It's like everyone did it in different areas. I wouldn't say that it really came from one specific place, but it's essentially when milk will be inoculated with lactobacillus and it separates and you get a cheese curd, and then you're left with the serum beneath it. And that serum is a potent probiotic source of lactobacillus, a whole bunch of different types of lactobacillus. This was then used to add to crops to boost health, prevent pathogens. It has a property that's called competitive exclusion. It's very good at competitive exclusion, which means that it will multiply very voraciously and outcompete different pathogens and other microbes that might be pathogenic or infect your plant. And so it was very commonly used. And this is like how cheese is made too. So anywhere that they were making cheese, they were making lactic acid bacteria serum, and then that byproduct was used as a fertilizer to fertilize crops and to promote nutrient cycling, to prevent pathogenic growth in the crops. And it was used all over the world. It's just an ancient practice.
Lactic acid bacteria, for short, people call it lab. Break down the steps on how to make it.
Yeah, for sure. So how it's taught in Korean natural farming is you collect rice wash, which is just starch that comes off of rice. But really, you can use any kind of carbohydrate source that will attract microbes from the air. I'm going to just go through the Korean natural farming process because it's the standard way to do it. But if you're in an area where you really insist on not going out and purchasing stuff, you can use any carbohydrate, that might be growing in your field or that's just local to your area because that's what you prefer. So you take rice, okay? What I like to do is I like to soak it overnight to really get all that starch softened off of it and shake it real good inside of a jar, probably like 50% volume of your jar and then the rest water. And you shake it up real good after about 24 hours, and it'll get all that starch off of it. And then you filter the rice out, and you're left with the rice wash. And then with that rice wash, you set it out in an area. People can do it in their house. They can do it, in a cupboard. I don't recommend that. I suggest you find a place outside underneath like a covered, like with like a little canopy, like bushes or whatever. Cover it with like a coffee filter or a paper towel, put a rubber band over it. Hope you don't have any wildlife or rodents that get into it. But put it in an area where local microbes are going to be existing, where you see that there's healthy plant life, right? Set it out there. Usually it takes only about 24 hours, but you wait until you see a thin film that collects on the surface of the water, and that is microbes. Those are microbes. It's just a whole bunch of bacteria that starts to grow on the top, and you know that now it's a source of bacteria. And it's not going to be just lactobacillus. It's going to be everything in your environment that's attracted to that carbohydrate and that profile of enzymes and carbohydrates. But it's enough lactobacillus. And what we do is we introduce it to a lactic acid environment to promote only lactic acid bacteria to multiply. And for, this example is milk. So people use milk. I like to get, if you're really bougie and you want to make like the best stuff, find some like local cream top, which will usually separate on its own, but adding more microbes to it just makes it that much better. You can definitely use any kind of milk that you get from the grocery store, 2%, whole milk, whatever. You can use whatever you have on hand. Getting something local is going to be super ideal. So you take 1 part your rice wash and you add it to 10 parts milk. I like to use a 5-gallon bucket. If you're making a whole bunch, you can use like a 30-gallon barrel, and that's enough for a whole field. So you mix it up. You're also going to cover that with like a paper towel or some kind of mesh, whatever. Oftentimes I just put the lid of the bucket on top of it, but I don't seal it because it's going to off-gas CO2, and we don't want to create a bunch of pressure inside the bucket and have it explode. So I don't seal it, but I just keep it kind of breathing. And then after about 2 to 3 days, you'll see it separate, and you'll have a cheese curd on top that's nice and thick. And I just cut it into pieces, pour it into like a paint strainer bag or something, like put a paint strainer bag in another, 5-gallon bucket, whatever, and pour everything in there, and then you just lift that cheese curd out. And that's cheese, and you can eat it, you can press it, you can make cheese with it and flavor it. And then we're left with the lactic acid bacteria serum. And that's the staple for the rest of the processes that we're going to be talking about. You can use it raw. People use it at 3.5 milliliters per gallon, or a ratio of 1 to 1,000. You can consume it. It's a little stinky, but you can do it. It's really good for your gut health. And that allows us to make fermented plant extracts. It allows us to make solid state fermentations, and then we can add it to things like Compost Teas or aerobic fermentations as well. Okay, so you mentioned the dilution rate 1 to 1,000 or 3.5 milliliters per gallon. So you're going to dilute it down. Is that going to, some people use it as a foliar, I believe. I mean, soil drench, is that happening? Oh, yeah. You can definitely use it as a foliar spray and introduce native biology, native lactobacillus species onto the leaf surface of your plants. And that would be the correct dilution for that too. The proper, dose would be like 3.875 or something like that. But 3.5 is just fine. You can use it as a foliar spray application. You can use it as a soil drench. I recommend doing both. It's really good for your plants, and just having it added to your foliar spray routine is really great. And just adding it to your soil drench regimen is also really good. I suggest using it about once a week, though. I wouldn't push it to every time you water because it can be very voracious. Like I mentioned, it's opportunistic. We want to maintain maximum diversity of biology in our soil, so we don't want to overuse lactic acid bacteria serum. Okay, and I heard that you can even drink this straight. Don't even need to dilute it. You just drink it. Is that true or no? You can do that. You can. I wouldn't. You can definitely do it. If you're you're really into that kind of stuff and you want to, you don't mind the very pungent smell and taste, you can absolutely do it. You can also dilute it down too. Humans, we secrete lactic acid. And so oftentimes on our bodies, we collect lactobacillus. And so the smell is very similar to body odor. So you could put that in your mouth if you want. But you can. You definitely can. Oh, that's funny. So that's lactic acid bacteria. I'd like to get into, you mentioned Bokashi composting next, or Bokashi, that as being a fermentation. This is something that I'm actually in the process of getting into. And so I'm super intrigued by this one. Break that down for me. So Bokashi is a solid state fermentation of any grain carbohydrate. And what that means is it's not suspended in water. We add a little bit of water to make it moist, spread the lactobacillus throughout the substrate, throughout the media, whatever grain carbohydrate we choose, which can be wheat, it can be oats, it can be a whole bunch of different things, right? It can be rice itself. And essentially what it does is it is an introduction of mucor, but it's also an introduction of lactobacillus, and it helps break down and compost things on the surface of your soil. So when you add Bokashi to the top of your soil, most of the nutrient cycling that's happening in soil is happening in like the first 6 to 8 inches of the soil. And so when you add a very, very potent inoculant like Bokashi, it really helps with the nutrient cycling in that area. And then that there's like a filamentous hyphae that comes out. There's been a lot of debate about what it is, but there has been confirmation through different doctors recently that it is mucor, and this is something that I've talked about in the past. It's in the Rhizopus family, and so what mucor does is it's in every single soil, and it plays a very crucial role in decomposition of nutrients. It also has the potential to create symbiotic relationships with mycorrhizae. So mycorrhizae kind of attaches to the root zone. It spreads out, it expands the surface area, allowing for more of the soil to be accessed by the plant, therefore more nutrients to be uptaken. The mucor will form a symbiotic relationship with that mycorrhizae also, and just extend that reach and that surface area even further. So it like multiplies the potential for nutrients to be absorbed by the plant. So let's get into the process of like how to make it, right? So you'll take your grain carbohydrate, you'll take your lactobacillus, you'll dilute your lactobacillus. I usually like to do 15 milliliters per gallon of the raw lactobacillus. But if you're using activated lactobacillus, which we can get into in a little bit, it would be about 30. And this is like what your FPE would be, or your AEM, your activated effective microorganisms. Dilute it to about 15 milliliters per gallon. And then if you want it to be just super simple, you can take your grain, you can mix it just like that. You mix it to what's called field capacity, to where it's moist, but it's not dripping. And that's like the maximum amount of water it can hold without runoff. Okay. Take that, you mix it very thoroughly. You remove all the oxygen from it. So I like to just put it in a trash bag. I think that that's the simplest approach. You just throw it all in a trash bag, you collapse all the air out of it, and you tie it. And then it's going to fill up with air, with carbon dioxide, because it's off-gassing a lot of CO2. And then after 3 weeks, it's finished. You dry it, and then you use it. It's very simple to make, and it's very effective in the garden.
This is something that I have been looking into, and there's recipes all over online. And, one of the ones I actually pulled up here, it has you start with 10 pounds of bran. And one thing I did research, where do you get it? We can get it at local feed stores. You can get it pretty cheap too. So you can get like huge, like 50-pound bags for pretty cheap. So that's usually where people start out with. I think I mentioned that one. And then you also mentioned using lab. A lot of the recipes call for EM1, which would kind of be a replacement. Would you say you could use either or in that case, or what?
So EM1 is a product made by Terragenics, and it is a lactobacillus ferment.
Got it.
So EM1, it's a consortium, right? So it has mostly lactobacillus in it, but it has other things like purple non-sulfur bacteria. It has some yeast in it. It has some other stuff too. You can definitely use EM1. That's going to be a little bit more expensive route. You can make your own, and it's significantly cheaper. Or if you'd like to go that route and you don't want to bother with the risk of, maybe messing something up, or the frustration of not having a successful culture, you can absolutely go out and get EM1.
Okay. And then there's also some other things listed here. You've got you added molasses and then water. And
yes, you can use molasses. So that's just like a sugar source. You can also add different fermentations. So let's say you had some FPJ, some fermented plant juice that you wanted to use. You could add some of that as a food source also. You could use an FPE that you've used this lactobacillus culture to break down different organic matter. You can use different polysaccharide sources. So these polysaccharides are just complex carbohydrates. So what I like to do is I'll add red beet juice, which has a very potent polysaccharide called inulin in it, which is really lactobacillus loves it. It's really great for lactobacillus. So I like to add red beet as my input. You can add different food sources. You don't have to. The grain itself is a food source, but if you want to really play with getting different cultures of biology and having different expressions kind of inside this Bokashi, you can definitely play with different food sources. You can use fruit juice, vegetable juice. People use carrot, beet, aloe vera, and those are kind of just different things that you can play with. And those ratios are going to be different depending on what you're going to be using. So I don't want to get too much into the specifics of that, because I don't want to lead anyone astray, because if you use multiple inputs, those ratios are all going to change as well. So using a food source like that is a good practice. You don't have to do it, but just the simplest form is just Bokashi lab, and letting it ferment for 3 weeks in an anaerobic environment without oxygen.
Okay, and then the end product, you kind of end up with like a dry bran, right?
Right. So you have to, like, after 3 weeks, you want to spread it out on like a flat, breathable surface. Some people have like mesh screens, like window screens and stuff that they use. And that's not much surface area, so you might want to get creative. Oftentimes people just put it on a tarp. That works just fine also. You just spread it real thin, ideally in a shaded area, because the sunlight can damage the biology. And you just let it dry out. If you're going to be using all of it right away, you don't have to let it dry out. You can just kind of like powder it up real good and just toss it right on the surface of the garden or the soil. You don't want it to be too thick, just like dusting it. You just want it to be spread kind of evenly. You don't want like a layer of it. If you've applied it to the point that it's like a cake, you've added too much. And also what I like to do if I do add a bunch is I'll scratch it into the soil surface to kind of break it up, because it can create a thick layer that can dry and then prevent oxygen going back into your soil. So you don't want to apply too much, because then it can create other problems. Also, don't add it to the stems, like around the stems of the plants. Sometimes if the plant is younger and it hasn't kind of developed its strength, you can add this strong microbial inoculant that's intended to break down organic matter to your stem, and it'll just break down the stem. So don't add it to the base of the plant. Like, keep like 3 to 4 to 5 inches, like a little donut around the base of the plant, ideally.
And then how often would you apply? Would you only apply it during a certain stage of the plant's growth? For example, flowering versus veg, or is it good throughout the whole life cycle?
I personally like to do it through veg. Keep in mind this is a potent microbe source, right? So if you have any kind of testing that you need to keep your microbial counts down, if you add this blooming, filamentous kind of like fungi to your surface, you're creating all these spores. Each little head has all these spores inside of it, and they'll fly around, they'll get everywhere. So if that's a concern to you, I definitely wouldn't do it. I like to keep it in like my vegetative growth stages or early flowering and then cut it out. But some people wouldn't agree with me. That's my approach.
Yeah, I can imagine some commercial facilities. I mean, they test for microbials. So it's like you mentioned, if you're using it in flour, it could potentially, they send out that for testing and they could potentially fail and then they have to throw away their batch. So
Definitely.
Totally get your reasoning for that.
And some people are allergic to it too. They're very sensitive to the spores being in the air. So if you're one of those people who's sensitive to spores being in the air, it's probably a practice that you.
Good to know. And so the Bokashi brand, after you make it, you can take that and not just apply it to the soil like you mentioned, but do a process called Bokashi Composting. And that's, I'd like to get into that if you've got knowledge on that. Cool.
Yeah, for sure.
I don't know if you want to start with kind of maybe how you make a Bokashi bin and then kind of using the Bokashi brand on the food scraps.
Yeah, we can talk about that for sure.
Okay.
This is definitely like a non-traditional way of composting. And it won't actually break the plant matter down like into a mush or into like a black substance like you would usually see doing like thermophilic composting. It almost like pickles it, right? Like kind of keeps it in this like pickled stage. But once it's taken out of the bin and you throw it in like a regular compost bin or on your soil surface, it'll break down very quickly. But in this controlled state in the Bokashi bin, it won't actually break down, and that's normal. And sometimes people see that and they get confused. So your bin is essentially a bucket or any vessel that has like a spout on the bottom. And the technique is you put down a layer of Bokashi at the bottom of it. You add a layer of your organic matter, usually like chicken scraps, kitchen scraps, and then you cover that again with Bokashi. And what it does is it'll create what's called leachate, which it'll kind of break it down. It'll create this juice, and that juice is very, very potent nutrient source that you can then add to your water and feed to your plants. You can add it to Compost Teas. It's rich in biology. It's rich in nutrients. And you essentially repeat that process of adding a layer of organic matter, of plant matter, adding a layer of Bokashi and doing it over and over and over and over again until you fill your vessel. And you let it break down. I'd let it kind of sit for like 3 weeks or so until you get all that leached out. You can even press it down to get more. And then what you're left with is plant matter that's still rich in organic matter, still rich in nutrient content. And you would break that up and top dress with it, and it'll break down very quickly. It's really great for your plants. You also don't want to make that too thick because it can create too thick of a layer that'll prevent oxygen from penetrating into the soil. Or you can add it to a regular compost bin and compost it, and it'll compost very quickly also.
Okay, yeah. Like I mentioned, I'm just kind of getting into it. So I have the 5-gallon bucket, and I also bought, it's actually used for car wash bucket. It's to get like a dirt trap. Basically, you put it in the bottom of the bucket, but it adds in the space. So that way the liquid can kind of beat down on that bottom part and spout on the side, like you mentioned. And then food scraps. I mean, you can put bones in there. You can put meat in there, right? All sorts of food you can put within there as long as you're putting the bran on there. At least that's my understanding of it. That's what I plan to do.
You can. I would personally avoid animal products.
Really?
Yeah.
Is there a specific reason?
I'm not a fan of flesh in compost. There's just, when you start playing with breaking down flesh and like cell and like animal tissue and stuff like that, you're attracting biology that's going to be breaking down flesh. And that's not oftentimes a good thing to have around us as animals. It can bring in pathogens and stuff like that. And while lactobacillus by nature is very competitive, like it can outcompete things, and the potential is much lower, the possibility is still there, especially if you make a mistake. And so I don't recommend using flesh and bones and stuff like that, because God forbid you get something that makes you very sick.
Makes sense.
Yeah.
So what would you recommend for somebody who is looking to like reuse food scraps or whatever? What type of things would you add in there safely?
Food waste. Your, lettuce, tomato, your plant matter, whatever if you're trimming things off of your plants, if you have, plant matter that you've defoliated or that you've, pruned the bottoms off of, that stuff is perfect. Or like the butts of whatever vegetables that you're not using. Vegetables, fruits and vegetable scraps is what I recommend using.
What about like bread or
you can add bread,
French fries or things like that?
So I would stay away from anything that's been like really heavily treated with pesticides and stuff like that, just because it's going to limit the microbial activity. You can definitely add carbohydrates for sure. But I think most ideally is going to be fruits and vegetable scraps. If you really want the bang for your buck, I would go that way.
And then that liquid that you mentioned that you would eventually go down, and then you've got the spout there, so you're draining it from the bucket occasionally. Does that need to be diluted before doing the application to the plants?
1 ounce per gallon is perfect.
1 ounce per gallon, okay.
Yeah. I wouldn't go any higher than that, honestly. It's pretty potent stuff. You can go lower than that, though. And if you do want to use things more, decrease the dosage. So if you want to increase the frequency, decrease the dosage. Increased frequency, decreased dosage. So if you do want to use it in every watering, you can drop it down to like 3 milliliters and go like that, because it's decent, and it's not going to be as potent as a lactobacillus source as straight lactic acid bacteria serum. So that's kind of an okay practice. If you want to do about once a week, once every 2 weeks, you can go up to about 30. But if you want to use it more, you have like a deficiency, you really want to correct quickly, I recommend smaller dosages.
And would that be for a foliar as well, or would you only recommend a soil drench with that?
You could use it as a foliar application. I would definitely drop the foliar application down a little bit lower too, to about 3 to 5.
And then, like you mentioned, that food isn't going to break down entirely, right? So you'll press it down, get all the liquid out, and then you'll empty out that bucket eventually. And I know a lot of people are, put it in their backyard and they'll bury it down in a hole in their yard, for example. So that's a way to go about it. And that's kind of the way I plan to use it here. I live in the desert. I have 1% organic matter in my backyard. And so I'm trying to reuse my scraps, and I have a regular compost pile. I have a vermicompost, but I want to continue to do new things here. So Bokashi really sparked my interest. There are things that you can't put in the worm bin that you can actually do through Bokashi composting. So I think that's what interested me the most is instead of just putting in the garbage, for example, I can put it into Bokashi composting and then eventually try to build up the organic matter that's in my backyard and be growing some fruits and veggies out there.
Yeah, 100%. And if you do feed it to your worm bins, the worms will eat it much faster too. And it's really good for the worm, like gut health.
Ah, okay. That's good to know. That's good to know. So you take it from Bokashi composting, big clumps, put it into your worm bin, it breaks it down even more. You get the worm castings in there.
And that would create a very high quality earthworm casting.
Awesome.
Yeah.
Cool.
So you said that, Bokashi can do some things that traditional composting can't. And you had mentioned like using meat and stuff. I kind of wanted to just take a little detour and kind of discuss ways of like composting meat, because I know that that's like a big thing for people.
Okay.
So black soldier fly larvae and like beetles are a great way to break down flesh. And so if you have a lot of food waste, let's say you're on a farm and you have, animals, you're a hunter and you have, a lot of flesh, whatever, that would just kind of, that you're not going to be using, whatever, you can create a black soldier fly larva bin, which are just like a species of fly. And they consume the flesh very, very quickly. Same with beetles, they'll consume the flesh very, very quickly. And then their excrement is used as a nutrient source and biological inoculant.
Good tip. Yeah, I appreciate you throwing that out there. Okay, so next up, you had mentioned an aerobic form would be Compost Teas. Can you get into that a little bit?
Yeah. So now we're going to tie everything back together. We can use all the inputs that we've discussed. The osmotic fermentation, the anaerobic fermentation. Actually, you know what? I think we should back up a little bit.
Okay.
Yeah, let's go to FPE, because we can talk about using labs and like molasses and food sources to then ferment into an FPE, and then we can use that as like an input and kind of wrap it all together.
All right, let's get into FPE.
So we talked about lactobacillus fermentation as a biological inoculant, right? But we can also use it just like when we do Bokashi buckets to break down organic matter to create available nutrients. This is also considered a solid state fermentation. But we can also do an anaerobic fermentation in water with lactobacillus to create what's called a fermented plant extract. And so what this is we collect plant matter. Typically, people will go out and they'll collect like fallen fruit. This is what I like to do. I like to go collect fallen fruit, peaches and apples. I also like to use kind of like gourds. I'll use like pumpkin and squash and melons and stuff like that, which have a lot of sugar inside of them. They also have a wide diversity, between all the things that I mentioned, including beets. They have a wide diversity of polysaccharides, which are different food sources, different complex carbohydrates, which will contribute different organic acids. And each organic acid, found in all of this diversity of plant matter, we have diversity of polysaccharides, we have diversity of organic acids. These organic acids will act as like plant stimulants. They act as phytohormones. Okay. And each organic acid will stimulate different processes in the plant, which is a whole other wormhole. We have things like jasmonic acid, which is the stress response hormone. We have auxins, we have gibberellins, we have cytokinins, which will all trigger different physiological effects inside of plants. So we add a whole different array of plant matter to water and we let it soak. It'll pull those organic acids out, and adding the lactobacillus will keep pathogens from multiplying. It'll also help break down that organic matter and release those organic acids, as well as release the nutrients inside of it. So what we do is we take the plants and the waste that we have around us, and we introduce it into water and lactobacillus and let it ferment, either with just the sugar sources that exist inside of the plant matter, or we can add a little bit of molasses also if we don't think we have enough sugar sources. And this gives us a very potent fertilizer that we can then dilute and add to our garden regimen. And it also is a source of phytohormones that we can then use to stimulate growth processes. And we can tailor the plant inputs used to have specific organic acid profiles to stimulate different growth processes in the plant during the time that it's needed. So these different phytohormones will have benefits for different plant growth processes. Like gibberellin is good for germination. Cytokinin is going to be good for like that transition stage. The ethylene is going to be good for like finishing and stuff like that. It's going to be good for your ripening stage. And so we can use resources online to look up different organic acid profiles. A good one is called Dr. Duke's Phytochemical Database. You can punch that into Google and you enter your plant into the search bar and it'll give you a full breakdown of your phytochemicals that exist inside of that plant matter. And then we can determine what we have in the plant that will stimulate which growth process in the garden that we're looking for. This can be kind of arduous because it's going to give you a list of like these organic acids that you may not know what they are. But you can like filter it by ppm, and then you can look up, the most potent sources in that plant to know like when I give this to my plant, when I harness these organic acids, it can promote certain growth processes in my garden. So that's what I really, really like to play with when making fermented plant extracts. Is like creating certain brews that are going to accomplish certain tasks and have specific profiles of nutrients that will complement certain growth processes in our garden as well. Let's say we have a plant that needs a particular amount of potassium at a certain growth stage or phosphorus at another one. We can tailor our fermentations for vegetative, early flowering, mid flowering, late flowering to help facilitate that growth process in our garden. And ultimately replacing a lot of products that, are out there on the shelf. Probably not all of them, though.
Okay, so fermented plant extracts, correct me if I'm wrong here, but what falls under that is fermented plant juice, fermented fruit juice.
So no, it doesn't actually.
It doesn't, okay.
No, it doesn't. So fermented plant juice is a osmotic extraction with like a sugar source, something that has a high osmotic pressure. That does not use lactobacillus. This is plant matter. I like to blend it to, keep the surface area, like as much surface area as possible. Plant matter, I like to make 55-gallon barrels of this, okay? You fill it a third to a half. If you're bold and you're really patient, you have a lot, you can do like 3/4. But I think like a third to a half is pretty solid. And then you add your lactic acid bacteria serum. The ratio is 1:1 to 20. So you add 1 part lactic acid bacteria serum, 1 part your food source, and then 20 parts water. So if you have a lot of sugar in your plant matter, you might want to tone down like how much molasses that you're using. If you have a lot of sugar, you can skip it completely, which is what I typically do. Is I'll fill it with a whole bunch of plant matter. I like using aloe vera, I like using beet, cantaloupe, squash, pumpkin, premature apples, stuff that I mentioned earlier. Blend it all up, add it to this vessel, which I actually have a video on this on my Instagram, talk about making a fermented plant extract. You add your 1 part lactobacillus to your 1 part food source to your 20 parts water. So let's say you have a 55-gallon barrel, right? Or we're just going to do it right. So we have a 55-gallon barrel. That's 213,125 milliliters, right? So if we divide that by, we have 22 parts total. We know we need about 3 gallons of lactic acid bacteria serum. A little bit less, about 2 and 2/3. We would add 2 2/3, 2.5 gallons of lactic acid bacteria serum. And if we have the volume equal to that in plant matter, probably a little bit more because, it's not all going to be sugar, maybe twice that amount in volume, we would then just add water to fill it all up. But if we're skipping the plant matter completely, you would just add molasses and fill it up with water. And this creates the fermented AEM, activated effective microorganisms. But with the plant matter, it is a fermented plant extract. When you fill it up to the top, you mix it nice and good. You cap it, you keep it anaerobic. You have to have a little valve, one of those little water valves, so like bubble ones, like that are used in fermentation. People use them in beer and wine and stuff like that to off-gas CO2. This also prevents oxygen from getting into the culture because it is an anaerobic fermentation. Lactobacillus is technically called facultative, which means it can survive in anaerobic conditions. But in this situation, we want to keep it anaerobic as possible. So we cap it, we seal it, we add one of those little air off-gassing bubble valve things. And we will let that sit for 3 weeks. And we watch it, don't open it. Let it sit for 3 weeks, test it with a pH pen, see what the pH is. If it's between 3.2 and 3.7, it's done. This will help us give an idea of how much lactic acid is inside of the culture. And when that lactic acid concentration reaches a certain level, it'll be within that range. And so we know that the bacteria, the lactobacillus, has released enough lactic acid to reach a particular concentration that we know that it's done. So 3.5, it's done. Anywhere between 3.2, 3.7, it's good. You can't get it down that low, it's a failure. It usually never gets lower than that, though. I've never seen that happen. You have to be careful when using plant inputs that are alkaline. If you don't get your culture low enough, the lactobacillus isn't going to thrive. So this is a problem when people are using the leaves of plants that they might defoliate the under part of. A common one that people do that with is often very alkaline. And so to kind of accommodate for that, we can use things like apple cider vinegar to drop that pH to about the 4.5 to 4 range, and it'll help facilitate the multiplication of the lactobacillus quicker. So it is good to check the pH before you start your culture and before you seal it, because if you're using plant matter that is very alkaline, it's going to interrupt the functionality of the fermentation. If you open it up after 3 weeks, pH is good, take a little shot glass of it. You know, there shouldn't be a pellicle, which is like a collection of lactobacillus that's going to protect the rest of the culture from oxygen. It's just like a thin film. If there isn't a pellicle, that's good. It means that your barrel was completely sealed and there was no air that got inside. If there is a pellicle, it's not horrible. It's not really a bad thing. It doesn't represent a fail, but there was oxygen that got in your barrel, and that's just something to note. Take a little shot glass, collect it, set it out for a couple days, see how quickly that pellicle forms. If the pellicle doesn't form, it was a fail. You didn't multiply lactobacillus. If it does, then that's a strong culture. This is a way of determining the strength and microbial density of lactobacillus-based products as well. A lot of people sell FPEs, a lot of people sell lactobacillus cultures, and if you don't have a microscope and you want to see if this thing is actually viable and alive, take a shot glass of it and leave it out. If it doesn't form a pellicle within a day or two, it's weak. If you get one, the quicker the better. Eventually it'll probably form something, but if you're at 7 days, that product is inferior to something that you could make at home. That's a good thing to know about. If you had a successful one, you got your pellicle, you got it within the pH range. This is something that you would use between 15 to 60 milliliters per gallon. I typically use it just at 30. You can use it as a foliar spray application. If you really want to get fancy with it, you can get a refractometer and see how much sugar content is in it. If you have a lot of sugar left in your culture after those 3 weeks, you overfed it. And it's not like a huge, big deal. You know, it's not going to create problems, but it will if you're going to start foliar spraying it. If you foliar spray sugar onto your leaf surface, you're going to start getting things like whiteflies or different pests that are attracted to sugars. So it's important to not overapply inputs that have a lot of sugar in them. And if you do, you need to have a microbial source along with it to help consume that sugar. But if you add too much sugar and you foliar spray it on your plants, you're going to have some big issues. So just to be mindful, it's better to underfeed your cultures than overfeed them.
I can't take notes fast enough. There's so much good information. So much new information for me. Like I mentioned, I don't really do ferments right now, but it is something that I hope to eventually get into in the future. So I'm learning, and I'm definitely going to be watching this episode back and taking notes here in the future. That's for sure. So you said it's different than FPJ and FFJ. Do you think now's a good time to get into those things here?
Sure, we can discuss the differences. I kind of covered it a little bit. Were there any specific questions or things that you'd like to know about regarding them?
Well, with FPJ, fermented plant juice, you're really only using plants. With the fermented fruit juice, using fruits. Can you use a combination of things was a question. Also, brown sugar, I know, is a thing that's often used. Would you recommend that to use for FPJ and FFJ?
Yeah, brown sugar is that's like what people.
The standard, okay.
Yeah, it's a standard, but it's anything with high osmotic pressure. I was chatting with some of my my nerd friends, we were talking about using dextrose because it's the highest osmotic pressure, like simple sugar out there, and it might pull out more. I've never actually done it, but anyway, yes, I would use brown sugar. And so you do a 1-to-1 ratio by volume, mix it up, kind of chop it, like masticate it a little bit, and then mix it with your brown sugar at a 1-to-1 ratio. And some people have changed the ratio. KNF kind of is known for changing their recipes over time, but 1-to-1 was what I was originally taught and what I have been using, and it works just fine. And then they let that sit for about 7 days and then collect the juices that have separated, and they'll be sitting on the bottom. And that's something that you use at a 1-to-1,000 application rate also. You need to have a microbial source when you're using it. Because like I said, it's pretty much just sugar. It's like syrup. And you can use it like syrup. You can eat it. You might want to seal it up with like a fermentation lid if you're going to be making it for consumption. But it's like sugar. And so when in KNF, people are using those inputs with lactobacillus, but they're also using what's called indigenous microorganisms, which is just the name of the input, which is it's rice that has been undercooked and left out in the forest to collect microbes. It'll form that filamentous hyphae layer like Bokashi will, like mucor. The claims are that it's collecting indigenous microorganisms and multiplying them. This is not a practice that I use. It seems to me that it's a method of introducing a mucor and biology throughout your soil. I just like to use Bokashi instead. I think it's simpler. It doesn't take a lot of time. Anyway, point being, you have to add a microbial source to your fermented plant juices. You don't want to add sugars to it. So what they'll do is they'll add IMO, they'll add FPJ, they'll add brown rice vinegar as your enzyme source. They add water-soluble calcium. They have like a whole range of things that they use, and they use them together. And it's important that you have that consortium of stuff. You have your inoculant, you have your enzyme source, you have your nutrient source, you have your protectors. They call them like the microbe police, which is the lactic acid bacteria. And they introduce it all together as this complete picture. And they recommend not using one alone. And I recommend that also. If you're going to be using something high in sugar, you have to have microbes and enzymes that go along with it. And that can be brown rice vinegar. That can be a little bit of apple cider vinegar. There are a lot of different enzyme sources out there. You can also use things like sprouted seed teas, which are seeds that have been sprouted, full of enzymes. You blend them. They're a very potent enzyme source. You can add, that too. But it's important that you have biology, the enzymes, the lactobacillus, and then your fermented plant juice, your sugar source, because it's just like straight sugar. And that can be problematic if used alone.
Yeah, you mentioned several different KNF processes, and that's a topic in itself, or an episode in itself, I should say. I had a couple people on in the past that were talking KNF, and yeah, it's so fascinating the way everything works together on that avenue. And yeah, we get super deep into that into a future episode.
Yeah, there's a lot to talk about around KNF.
But maybe we should stick to the fermentation. One thing I would like to get into, if I think we have time here, is the compost teas. So I know a lot of people use compost teas. There's right ways to go about it. There's wrong ways to go about it. There's some people that say that you shouldn't use them at all. So maybe that's the question I ask you to begin, is this broad question. You have folks who say they use compost teas frequently and they gain a lot of value out of it. You've got other people that say, just use compost, just top dress the compost and water it in. Doing a tea isn't going to have any additional benefits. What's your take on that?
So, it depends. Yeah, adding compost, top dressing compost is going to be if the compost is quality, is going to be a much safer way of introducing biology and nutrients. It's in its original form. You add the water, the microbes get introduced, the nutrients are there. It makes it very simple. Where the problem comes in is if you have a lot of soil, you need a lot of compost to cover it. So if you're growing in a field, you're growing any significant amount of food, and you don't have enough compost to do that, compost teas are a great way to stretch that biology. And oftentimes, people don't take into consideration that when we're making compost teas, we can also add other things to the compost tea that wouldn't be present inside of a thermophilically composted compost, or even a mesophilically composted, like insect frass or earthworm castings. A little bit earlier, I had mentioned diversities of organic acids used as like phytohormones to promote plant processes. We can add inputs that introduce those organic acid profiles to our compost teas and make them more effective and more readily available to our soil. We can also use different inputs in our compost teas to encourage microbial multiplication. All cells require amino acids. All proteins, are formed through amino acids. So what I like to do in my compost teas is I'll add an amino acid source. I'll add different diversities of complex carbohydrates, of polysaccharides, to encourage diversity of biology. Compost teas are not the native environment for soil biology. Sure, soil gets wet, but it is not water, right? So when you make compost teas, you're taking biology out of its native environment and you're putting it in water, and you're going to limit the diversity that it can multiply. It still is very effective, but you're not going to have the microbial diversity that you would get if you were to just top dress it. But top dressing an acre is a huge task. Top dressing, a half acre, quarter acre, whatever. If you're trying to bioremediate a piece of land, it's going to be very difficult and take many, many years to do composting traditionally, and adding compost to the soil surface. Compost teas are a great way to just help kind of bridge that gap, to add biology, to help cycle nutrients earlier. So I think that they're just tools. And I think to be like, oh, this tool is just better than this tool, I think that this tool has an application specifically that's going to be ideal. And then this tool has another. And all of these things that we're talking about today, they're all just tools. And some of them are going to have strengths, and some of them are going to have weaknesses, and some are not going to be ideal in certain situations. Compost tea is just another one of them. So you can play when making compost tea. People use compost, obviously. You can use thermophilic, which is going to be your traditional turned compost. You use mesophilic, which is going to be your earthworm castings or your insect frass. They're going to have different profiles because they are made through different processes. One is through the turning and heating up of microbes that break down the plant matter into compost. The other is being consumed by insects and going through their gut biome and using their excrement. I prefer mesophilic breakdown, mesophilic composting, because when it doesn't get to that higher temperature, it isn't going to cook off other biological species that can't survive in those temperatures. Microbes, some of them prefer acidic environments, some, more basic, some, colder, some hotter. And as we heat them up, we're creating an environment that those microbes that want that warmer temperature are going to exist in. While in a mesophilic, we're going to leave a larger profile of biology. So in my compost teas, I like to use earthworm castings specifically. And it reduces the potential for pathogens if whoever made it didn't turn it properly. Making compost is an art, and if you mess it up, you can introduce pathogens. I've seen people introduce Septoria and Fusarium, different pathogens through thermophilic compost very regularly, especially with mushroom compost that people use. So I like to use earthworm castings as my biological inoculant and my compost source. So I'll take earthworm castings, I'll throw them in a vat of water, your brewing vessel. People will like to add bubble snakes, which is just like a tube that wraps around the bottom that has holes in the bottom, and they blow bubbles down and it agitates the water. And you have a brew bag, and you put all your inputs in it, and it kind of agitates it, and it scrubs the biology off of your inputs, and it soaks into the water. And then while it's in the water, it has your food source, which people usually like to use molasses. I like to play around and add some different things. I like to add oat flour, which is something that Dr. Elaine Ingham talks about. It has more lipids in it, and there's more like, fats in there for you and for the biology. And it has a lot of starch and stuff, and it has, polysaccharides that are good, different complex carbohydrates that'll feed more diversity of biology. And then I like to use beet juice, which you can see, which I've talked about, which is inulin. It's a very strong prebiotic. These polysaccharides, they're prebiotics, right? And that's what I make my teas with: oat flour and beet juice with earthworm castings. And then before the long fermentation process, which is usually about 24 to 36 hours, I like to add things that are going to promote cellular development. Things are going to promote microbial multiplication. Like I said before, all proteins require amino acids to be built. So I like to add amino acids. You can get products that have amino acids in them. There's soy-based amino acid products. There's fish-based amino acid products. You can use aloe vera, which I also like to use. However, I prefer to use it at the end, and I'll get to that in a second. So I add an amino acid source, but then I'll also add a calcium source. There is a Korean natural farming input called water-soluble calcium, and it's essentially exactly what it says it is. But it's calcium carbonate that has had apple cider vinegar added to it, and it breaks it down and it makes it an available calcium form. It makes calcium acetate, because apple cider vinegar has acetic acid in it, and it creates a chemical reaction. Calcium acetate is one of the most available forms of calcium that we can find in nature, and I'll add that to my compost tea as well. So I'm creating the foundation for microbial multiplication and diversity of biology in my compost teas with a biological input, earthworm castings, which has a very wide range of biology. Compost teas can be as creative or as simple as you want them to be. You could literally throw earthworm castings in a bucket with molasses and let it sit, and then dilute it and water it in, and it would work. You can get very creative with what you're using. You can go find different, plant inputs that have different sugar profiles, that feed different profiles of biology. Which is what I like to do. Which is what I'm kind of known for. Is kind of creating these more concoction, potion-based compost teas to use in my garden. And then there will be some inputs after you brew that you would want to add, that you don't want to add beforehand. For me, that's your inputs that are going to have rich profiles of organic acids or amino acids that you don't necessarily want to be broken down. So I like to add my aloe vera, which has 19 of the 20 different amino acids. It has a whole bunch of enzymes. It has a polysaccharide called acetamenin, which has tons of soil benefits, and will elicit plant responses, just that compound itself. It also has auxin in it, which is a plant growth hormone, cause explosive growth in the plant. I don't want any of those compounds to be broken down by biology, so I'll add them at the end. So if you have a plant input or just input in general that you want to use to stimulate specific growth processes, add it to the end of your compost tea, because biology changes compounds from one thing to another. And so when you're using an aerobic fermentation, that's much more likely to happen than in an anaerobic one. Also, lactobacillus, lactic acid bacteria serum, since I talked about how voracious and opportunistic it is, if you add it to your compost tea pre-brew, it can multiply and dominate the rest of the biology in the brew, leaving you with less diversity. So I like to add that to the end also.
Good to know about the inputs added after the brew. I don't think a lot of people add inputs after the brew, to be honest. Now, you mentioned a brew time of 24 to 36 hours, and there's a lot of information. Some people say it should be shorter. Some people say it should be longer. Can you just give us high level, what are the risks of brewing for too long? And then what are the downsides of brewing for not long enough?
So here's the deal. The time period thing is just to make it easy for people. It's going to depend on a lot of different factors. How much you fed, how much inoculant you used, what the temperatures are outside, what the temperature of your water is. Does your water have any kind of contaminant? What's the calcium amount? What's the sodium amount? What's the, do you have chlorine in your water? Which always, you should not have chlorine in your water, by the way. Good point. These are all going to affect how quickly the biology is going to be multiplying in your brew. So when people are like, do a short brew, it's because like, yeah, if you mess it up and, it multiplies too much, you can have an imbalance in your biological profiles. Things like protozoa will start to take over, which can be potentially pathogenic. They're also an important part of nutrient cycling. They're like our predators. I know that's complicated, but there's many different species of them. So in like soil food web sciences, right, we use microscopy to identify the profile of biology existent in our compost. We have a particular microbial density of lower bacteria, and then we have our protozoa, we have our amoebas, we have different types of protozoa, we have our ciliates, we have our flagellates, we have our rotifers and stuff like that. And there's things that we do and that we don't want. And given a certain amount of time, we're going to see our protozoa show up in numbers, which will consume a lot of our lower bacteria, which is what we need. But we also need a certain count of protozoa. So it's one protozoa, usually a ciliate, per 10 field views. And so when people are like, how long should I brew my tea? And it's like, well, it depends on how many protozoa have multiplied inside of your compost tea. So it's not a simple answer. The simplest, safest way would be just like, do it for 24 hours. Do it for 6 hours. Do it for, just mix it and water it in, because we're playing with biology. We're playing with bacteria, and there's a reason why people are cautious when playing with bacteria and multiplying bacteria. It's because you can mess it up. You can make mistakes. You can make people sick. You make yourself sick. That's one of the reasons why people say don't do it. This is one of the reasons why I only use earthworm castings. I had a buddy who's turning mushroom compost. He got some like fungal infection in his blood. It's just we're farmers. We play with plants and stuff. We play with biology, but like I just had someone yesterday tell me how he got something from turning compost. You can have like aspergillus. You inhale aspergillus, like you're gonna make yourself sick. So to answer your question, there isn't a very simple answer. I usually do 24 to 36 hours. 24 if you want it more bacterial dominated, and 36 if you want it more protozoa. And to determine which one you would want, you would need to get a microscope and examine the profile of biology in your soil. And so if you have low microbial density, you want to add a compost tea that's going to have a lot of lower bacteria in it. It's going to have like a lot of simple bacteria. And then if you see in your soil that there aren't enough protozoa, you're going to want your compost tea to run a little bit longer to introduce protozoa into your soil. And that's only determined by with a microscope, through microscopy. So that I do 24 to 36 hours.
Understood. Well, there has been so much good information in this episode as well. Thank you so much for coming on to the podcast again. Let's wrap things up here. Can you tell the listeners how they can find you? And if you have anything upcoming in the future, now would be the time to spill it.
Yeah, for sure. So you can find me on Instagram at Luna All Day, LunaLlday. I am a breeder. I have stuff that I've bred specifically for large yields, unique expressions, unique aromas, and stuff like that. I have a Patreon. If you go to my Instagram, you can check out my Patreon where I have a whole bunch of different recipes like the ones I was talking about. I go really into depth into the sciences behind this stuff. I have tons of research papers, discussions about, all the different things. I have like 100 different channels covering different topics like we did today, and breaking them down in detail. And you can interact with me and stuff too. I do podcasts with the FCP. I took a break for a while, but I'm getting back on it. And I should have, I'll be doing episodes every Friday at 5:00. You can find that on YouTube. I've been working with Skunk Magazine. I'm published in Skunk Magazine. We just had a new skunk seed line drop. Our website is up, skunkglobalgenetics.com.
Awesome. Well, I'll definitely have a link, I believe, to your link tree. That's, I think, what we used last time. So I could probably do the same thing this time around. That'll get all the links to what you just mentioned. I got to give a shout out to all the Patreon folks that support this podcast, patreon.com/MrGrowit. 100% of what is donated there goes right back on the podcast. So thank you so much. I'm going to put on the screen right now the folks that are in the producer tier. Special thanks to you. Luna, once again, this has been just an awesome talk. I would like to invite you back on for a part 3, but I think I would like to talk about breeding. You said you had 10 years of breeding experience. There's so many different ways to go about breeding. I would love to sit down with you at some point in the future and talk breeding if you're down.
I'm absolutely down.
Awesome.
Yep,
all right. Well, we will leave it at that. Once again, thank you so much and hope you enjoy the rest of your day.
Thank you. Have a great day.
Peace out, everyone. Catch you in the next episode.